Dear friends, another audio fragment of NMEICT project, an initiative of MHRD. Introduction to nucleic acid and proteins. Students will start with the nucleic acid. Nucleic acids were discovered long back in the year 1869 by Frederick Miescher. Almost after 90 years, an American biologist, Watson, and an English biologist, Crick, combinedly proposed the double helix structure of DNA. Nucleic acids are the building blocks of all living organisms. Friends, we know nucleic acids allow organisms to transfer genetic information from one generation to another. Commonly, there are two types of nucleic acid: deoxyribonucleic acid, which is better known as DNA, and ribonucleic acid as RNA. DNA is found mainly in the nucleus of the cell, whereas ribonucleic acid, that is RNA, is found mainly in the cytoplasm of the cell, though it is synthesized in the nucleus. When a cell divides, its DNA is copied and passed from one generation to the next generation. So, can we call DNA as an informational molecules? Yes, because their primary structure contains a code or a set of directions by which they can duplicate themselves and guide the synthesis of proteins. Or, in more simple words, you can call it as a programmatic instructions for cellular activities. When organisms produces offspring, this instruction in the form of DNA are passed down, and RNA is involved in the synthesis of protein. So, together with proteins, nucleic acids are the most important biological macromolecules. Each is found in abundance in all living things, where they combinedly functions as encoding, transmitting, and expressing genetic information. All nucleic acids are made up of same building block, that is nucleotides. These nucleotides are made up of three parts which are covalently bonded together. These three parts are the very important one, pentose sugar; the second one, a base that has a nitrogen atom; and the third, an ion of phosphoric acid known as phosphate. But when a base and sugar is combined, it is called nucleoside. Which are the sugars of nucleic acid? The first important component of nucleic acid, 5 carbon containing monosaccharide, we call it as pentose, are found in the nucleic acid structure. RNA contains D-ribose, while DNA contains deoxyribose. Ribose and deoxyribose differs in structure at carbon 2 of the pentose ring. Second component of nucleic acid, that is base. The second important component of nucleic acid, bases. The nitrogenous bases found in the nucleotides are aromatic heterocyclic compounds. These bases are of two types, purines and pyrimidines. Purine has both a 6 and a 5-member ring, but when you see pyrimidine, it has a single 6-member ring. Purines are named in the anti-clockwise direction, while pyrimidines are numbered in the clockwise direction. Cytosine, thymine, uracil are the types of pyrimidine, whereas adenine and guanine are the types of purine. Although both purine and pyrimidine rings have one 6-membered components with 2 nitrogens and 4 carbons, the purine and pyrimidine are not related metabolically. That means they have a different pathway for the biosynthesis and the degradation. The third component of nucleic acid, phosphate, a major requirement of all living things. A major requirement of all living things is a suitable source of phosphorus. One of the major use for phosphorus is as the phosphate ion, which is incorporated in DNA and RNA. So all these three components make nucleotides. But how this nucleotide polymerize to form DNA? A strand of DNA or RNA contain nucleotides which are covalently attached to each other in a linear fashion. The sugar and the phosphates in the nucleic acids are linked to each other in an alternating chain, which we call it as a sugar-phosphate backbone. The bases project from this backbone, and the backbone is negatively charged due to the negative charge of phosphate groups. Second important structural feature is the orientation of the nucleotides. The carbon to which phosphate group attaches are the 3' and at the 5' carbon is attached to the sugar. So, the ends of nucleic acid molecules are referred as 5' and 3' Now let us see the famous Watson and Crick model of DNA. James Watson and Francis Crick combined the physical and the chemical properties of DNA with the feature of X-ray diffraction pattern suggested the helical structure of DNA. Let us go through the important features of the DNA molecule as defined by Watson and Crick in the year 1953. The first important feature of this model is two strands are coiled about one another to form a double-stranded helix. Second feature is purine always opposites a pyrimidine. Third feature, the two bases in each pair lie in the same plane and the plane of each pair is perpendicular to the helix axis. So, these base pairs are rotated at 36 degrees with respect to each adjacent pair. So, you can say that there are 10 pairs per helical turn and the diameter of the double helix is 2 nanometer. Another feature is two external helical grooves are formed. A deep wide one, which we call it as a major groove, and a shallow narrow one as minor groove. Another feature, chain held together by hydrogen bond. For example, guanine is paired with cytosine by 3 hydrogen bond and adenine is paired with thymine by 2 hydrogen bond. Another important feature of the model is antiparallel orientation of two chain. That means one chain in 5' to 3' direction, antiparallel orientation of two chain. That means 5' to 3' direction and 3' to 5' direction of the chain. Overall, the molecule is stabilized by large number of hydrogen bonds and hydrophobic bonding between the stacked base. Students, it seems from the above discussion that DNA is only of one type, but it's not so. There are different types of DNA. The DNA double helix can form different types of secondary structure. The primary difference between the form is the direction that the helix spirals. aDNA, bDNA, cDNA and zDNA. Basically, they are divided into two groups, A, B and C. They are having right-handed helix, whereas Z has left-handed helix. Among all these four, B is the predominant form that is found in all the living cells. Under certain conditions, the two strands of DNA can twist into aDNA and zDNA, which differ significantly from the bDNA. In addition, the helical backbone in zDNA appears to slightly zigzag as it winds itself around the double helical structure. The numbers of base pairs at 360 degrees centigrade turns are 10.9, 10 and 12 in aDNA, bDNA and zDNA respectively. In bDNA, the bases tends to be centrally located and the hydrogen bonds between base pairs occurs relatively perpendicular to the central axis, whereas the bases in the eDNA and zDNA are substantially tilted relative to the central axis. The ability of the predominant bDNA to adopt aDNA and zDNA conformations depends on the conditions. For example, aDNA conditions occurs under the conditions of low humidity, whereas zDNA is at different ionic strength medium. For example, at high ionic strength, formation of zDNA conformation is favored by a sequence of bases that alternates between purines and pyrimidines. But if you take a lower ionic strength, the methylation of cytosine bases can favor zDNA formation.